All right. So today we're going to be doing the Solving Mystery Molarities using Principles of Osmosis lab. So to get us started, I'll give you a little background information and then we'll jump into the procedure. So I've made 6 different sucrose solutions here. They are of different varying molarities. We have up into 1 molar sucrose and then going down to 0 molar sucrose, which is just going to be pure water. And then we have different molarities in between. You can find those values in your lab handout. Your job is going to be to use the principles of osmosis and water potential to figure out which solution is which molarity. To do this, you need to make sure that you go back and read some of the introductory information about water potential. This is going to be super helpful in terms of figuring out if water will move into our cells or out of our cells based on the molarity of the sucrose solution that the cells are sitting in. Today we'll be using potato cells to simulate this process because potato cells are plant cells, and so we'll be able to actually talk about the fact that pressure will build up inside of those cells as water moves into the cells. On the flip side, if water were to move outside of those cells, you would start to see those cells kind of shrivel up, which is what we see in plants when they start to lose water. We start to see them kind of shrivel up and tilt over a little bit. So what I'd like you to do right now is to go back and read the introductory information in your lab handout and read the information about water potential and how it's calculated before you move on to the procedure. Now that you're at the procedure, what you're going to want to do is just follow along with my process. I already set up this lab yesterday because you'll see in your procedure it says it needs to sit overnight. So I'm going to show you how we do the initial setup, and then I will give you some information that I took yesterday, some of the actual masses that I took yesterday, and then together we will do the procedure part that occurs after the overnight part, and you'll be able to use that to do all of your calculations. So if you look here at my lab setup, you'll see I had a potato that I cut into kind of these cylinders. I'm going to use this potato to create what I'm going to call potato cores. And you can see here that a potato core is like this little cylinder of a potato. The easiest way to do this is with a tool called a corer. And all I did to get these potato cores is I just kind of pushed it through my potato. And once I push through enough, I can just kind of pull them right out the top. So I already did that for you guys, so you don't have to watch me core 18 potatoes. But that is how that occurred and where you see these little cylinders came from. Our next step in our procedure tells us that we need to find the initial mass of the potato cores. We're going to be creating 6 different cups because each cup is going to have one of these different solutions put into them. And so we need 6 sets of potato cores. I'm going to use 3 potato cores for this lab. You could really use any amount because all you're going to be calculating is the difference between the initial masses and the final mass. So it really doesn't matter how many you use, but 3 seems like a pretty easy number, so that's what we're using. In this piece of the procedure, what I would do is I would take my group of 3 potato cores and I would place it on my scale. And I'm going to get an initial mass that way. I'm not going to read you the mass that I see in front of me right now because your initial masses that you use are going to be from the potatoes that I already took the initial mass of yesterday. So I'm just going to show you what I would do once I took the mass. So I'd record the mass of these potatoes and then I'm going to put them into their cup. I'm then going to cover it with the corresponding solution. So you'll see on your lab that it says like red, orange, yellow, green, blue, and purple. So I'm going to start with red, and I'm going to put just enough sucrose solution in here to cover up those potatoes. I'm going to put a lid on it because I would be leaving this overnight, and I don't want any type of evaporation to occur that might kind of mess with my results. I'm going to go ahead and do that for the rest of my potatoes. So again, I would take the mass of these now. I'm going to skip that step here because I already took the initial masses of the ones that we're using for our lab. And I would cover this one with the orange. Remember that you have 6 different concentrations of sucrose solutions in your lab today. They are not in the order of these colors as I have them set up here. So red is not necessarily the highest concentration, and purple is not necessarily the lowest. I have kind of mixed them up. I was very careless when I made my solutions, and I forget which one is which, so you're going to have to figure that out. So I have my red, orange, and yellow so far. I'm going to go ahead and set up my green. This is what you would be doing if you were doing this lab with me in class. You'd set this up one day, we'd let it sit overnight, and then we'd come in the next day and we'd see what happened to our potatoes. So that's what we'll be doing in 1 minute once I get these ones going. Blue. And finally, we have purple. All right, so I have my 6 setups now ready to sit overnight. About 24 hours probably would be the best time if you're gonna actually go ahead and do this lab in the future. But what we're gonna do is we're gonna take these ones, put them to the side, and then I'm gonna grab the ones that I set up for you yesterday, and those are the ones we're going to take the masses of. While I get that going, what I'd like you to do is take a look at the table that you see on your screen, which will show you what those initial masses are. You're going to want to pause your video and then record those initial masses in your table on your lab handout, so that then when we come back and get the final masses, you'll be able to use those to compare how the mass of those potatoes changed overnight. All right, so now you should have recorded all of your initial masses, and I've pulled out the ones that correspond with those initial masses that you wrote down. And these have been sitting since yesterday afternoon. And now what we're gonna do is figure out what the final mass is for each of these groups of potatoes. So I'll start with red and work my way down, so it should match up with your table perfectly. So I'm gonna take my red ones out. If you look at your procedure, it says to kind of blot the potatoes dry a little bit. And so the reason we're going to do that is we don't want them soaking in extra water, because this will kind of make the mass higher than what it really is. We just kind of want to know, did my actual potatoes themselves gain mass, lose mass, or maybe they stayed exactly the same. So I'm going to take these out. They're just potatoes, so I touch them with my hands, it's fine. All right, and I'm just going to kind of let them sit there for a second to kind of dry off a little bit. And then I'm going to place them on my scale. And my scale tells me that my final mass for my red potatoes is 1.07 grams. So I'm going to go ahead and write that down. I'm going to take these off and move to my orange. I'm going to have some beautiful rainbow colored hands at the end of this. All right, my orange potato final mass is 1.61 grams. As I'm taking these yellow ones out, I'm noticing that they are kind of shriveled up a little bit. And I would like you to think, while you're waiting for me, to maybe get an idea of what does that mean in terms of, did water move into the potato cells or out if they are all shriveled up? Their final mass is 1.21 grams. Next I have my green, and I'm making another observation here for you guys. My green ones look as though they have maybe gotten bigger over the night. And so I would like you to also think about, does that mean that water might have moved in or out of my potato cells? So my green final mass is 2.03 grams. Next I have my blue ones. These ones also look to be maybe a little bit shriveled up, similar to the ones in the yellow. And their final mass is 0.93 grams. And finally, we have our purple ones. Their final mass is 1.37 grams. So now you should have filled in your table. You should have your initial masses and your final masses. You're going to be doing a couple of calculations before you are able to make any conclusions. Your calculations, what you should be doing is finding the actual mass difference, so the change in mass. Remember, this could be positive or negative. Don't get rid of your positive and negative signs here. It's very important when we go to do our conclusion that we know, did my potato gain mass or did it lose mass? So pay attention to the positive and negative signs there. You're then going to be figuring out the percent mass difference. The percent mass helps us to kind of calibrate all 6 of our potato samples to one another, because they didn't all start at the exact same mass. So we don't really care about the raw mass that they gained or lost. We care about what was the change in the percentage. This helps us to actually be able to compare them to one another. Once you get your percent changes, you're going to be using that information to figure out which molarity goes with each color. To help you with this, I want you to think about the words hypertonic, hypotonic, and isotonic that you have learned about, and think about, in terms of the solution, what would make water move into the potato cell, or what might make water move out of the potato cell? What type of solution would the potato cell have to be in order for water to leave it, or in order for water to move into it? Once you have those lined up and you know which color goes with which molarity, you're then going to be creating a graph for me and answering a couple questions about water potential. It's very important that you get your percentages and your percent change in mass accurate, so that you will be able to figure out what the actual molarity of a potato cell is. We're going to be using these concentrations to help us figure out what is the molarity of a potato cell, which should be exactly where we would expect no change in mass, right? Because if the molarity of the potato cell is equal to the molarity of the solution outside of it, we wouldn't really expect any change in the mass. We would expect them to be at equilibrium. So go ahead and do your calculations. You will use those to create your graph and to answer your analysis questions, and then we'll be all set.